The mammalian nervous system is organised structurally into two systems. The central nervous system includes the brain and the spinal cord. The peripheral nervous system includes nerve cells and connects the CNS with the rest of the body. The peripheral nervous system is divided into somatic and autonomic nervous systems. The somatic nervous system is made up of cranial nerves, sensory and motor neurons. The autonomic nervous system is in control of automatic functions such as the heart rate and breathing, as well as playing a role in reflexes. Within the autonomic nervous systems, there are two other systems known as the sympathetic and parasympathetic systems. When a fight or flight response occurs during an emergency, the sympathetic nervous system is activated. This leads to changes in the function of many bodily systems and organs, including an increase in heart rate. In the lungs, bronchial muscles relax, allowing more air in, and along with an increased heart rate, this allows more oxygen to be pumped around the body to respiring tissues. This is important during a fight or flight response, as muscles are expected to contract, allowing you to escape the danger. In response to the activation, the sympathetic nervous system also has a number of metabolic changes. Digestion of food is decreased in both the stomach and small intestines. The salivary glands produce less saliva, hence in times of stress we found that our mouths may go dry. Other metabolic changes include decreased urine secretion. Neurons contain nerve cells that send messages through chemical messengers and electrical impulses, also known as action potentials. During rest, nerve cells are polarized with a value above about -70 millivolts, known as the resting potential. During rest, nerve cells are polarized with a value of about -70 millivolts, known as the resting potential. The value is mostly maintained by sodium-potassium pumps, creating an electrochemical gradient. In this mechanism, 2 sodium ions are actively transported out of the cell, while 2 potassium ions are transported into the cell. This creates an overall positive charge in the extracellular space and an overall negative charge in the intracellular space within the axon. Once the nerves are stimulated enough to reach the threshold of -55 millivolts, the sodium channels on the dendrites open. This causes voltage-gated pumps to open, moving sodium ions in and potassium ions out of the cell, leading to depolarization. Once the threshold of positive 40 millivolts is surpassed, the cell begins to repolarize as the voltage-gated sodium channels close and voltage-gated potassium channels open. This brings the cell back to a negative resting potential, ready for the following action. Hyperpolarization is caused by the slight delay in gates closing, meaning the cell can become too negative. To counter this, the resting potential is re-established by the sodium gates opening. The other advantage to this stage, known as the refractory period, is preventing another action potential to be stimulated or generated whilst the sodium channels are recovering. Dendrites receive the electrical impulses, which are then transported down the axon, where they bounce from one rodent to another, allowing for efficient transport.
When the autonomic nervous system is stimulated by a jump scare, it triggers various physiological responses in both the visual and auditory systems. Visual responses to the jump scare involve changes in peripheral vision and pupil dilation. The autonomic nervous system, specifically the sympathetic branch, becomes activated in response to a sudden and startling stimulus. This activation leads to an increase in peripheral vision, which means that the field of vision expands, allowing you to notice your surroundings more effectively. Simultaneously, the autonomic nervous system triggers the dilation of the pupils, a process known as mydriasis. The dilation of the pupils allows more light to enter the eye, resulting in improved vision. By dilating the pupils, the eyes can gather more visual information, enhancing visual acuity and the ability to perceive details in low light conditions. These adaptive responses prepare the visual system to gather as much information as possible in response to the sudden visual stimulus of the jump scare. In terms of the auditory response to the jump scare, it involves the conversion of sound vibrations into electrical impulses that are transmitted to the brain. The auditory system contains a spiral-shaped structure called the cochlea, which plays a vital role in processing sound. The cochlea is divided into 3 fluid-filled compartments, and when sound vibrations enter the cochlea, it causes the fluid to move and stimulate the hair cells which are located along the cochlear walls. The vibration patterns is then converted into electrical impulses by the specialized sensory hair cells. The electrical impulses generated by the hair cells are transmitted to the brain through the 8th cranial nerve, also known as the auditory nerve. The auditory nerve carries these electrical signals to various regions of the brain responsible for auditory processing, including the auditory cortex. Similar to the visual response when the autonomic nervous system is stimulated by the jump scare, the ears perk up. This response is characterised by heightened auditory sensitivity, allowing for sharper hearing. This increased sensitivity enables the auditory system to detect and process sounds more efficiently, enhancing the ability to perceive and analyse the sudden auditory stimuli associated with the jump scare.
In our body's response to stress, the sympathetic nervous system triggers the fight or flight mode, releasing a powerful chemical known as adrenaline. Atop our kidneys sit the adrenal glands, with an outer cortex and an inner medulla. The sympathetic nervous system signals for the medulla to release adrenaline into the bloodstream. Adrenaline, a hydrophilic peptide, binds to adenal cyclase on the cell membrane. This initiates the conversion of ATP into cyclic AMP, a second messenger that triggers various cellular responses. Cyclic AMP binds to proteins, setting off a cascade of effects. For example, it activates protein kinase stimulating the liver to release glycogen, providing an immediate source of energy. Adrenaline's effects range from increased heart rate to heightened awareness. It redirects blood flow, relaxes airways, and temporarily suppresses digestion and immune function. Importantly, this heightened state is a temporary survival mechanism, and the effects gradually subside, returning the body to normal. Prolonged exposure can have negative health effects, so the body carefully regulates this response for balance.
We will now dive into the intricacies between our heart and lungs. Picture this: adrenaline, the body's natural alarm system, igniting a cascade of responses. Specifically, it binds onto beta receptors on the heart, known as beta-adrenocaptor subtypes. The heart then responds with a vigorous beat, pumping at an accelerated rate. This ensures a higher cardiac output, delivering a surge of oxygen, glucose, and nourishment to our skeletal muscles. This heightened activity is crucial for the body's swift response. However, adrenaline-induced changes don't stop there. Systolic blood pressure rises while diastolic blood pressure drops. The isometric contraction phase shortens and the speed of contraction increases. Adrenaline also orchestrates vasodilation in blood vessels, leading to skeletal muscles and the digestive system. Imagine the muscular layers relaxing, widening the vessels. This is to optimize blood flow. More blood rushes to the digestive system, supporting the breakdown of glucose for our hard-working muscles. Now let's take a look at the respiratory response to fear. Fear triggers the release of adrenaline, stimulating intercostal muscles through beta receptors. Result: a rapid increase in breathing rate, ensuring our bloodstream is flooded with oxygen. In just 60 seconds, adrenaline transforms our physiology, preparing us to face challenges head-on.
When a nerve impulse is sent down the motor neuron to the muscle cell, it travels towards the individual muscle fibres. As the nerve impulse reaches the muscle fibres, acetylcholine is released, leading to the depolarization of the membrane, known as the sarcolemma. This interaction occurs at the neuromuscular junction, the site where the nerve impulses communicate with the muscle fibre. From the neuromuscular junction, the impulse continues its journey by travelling down the extensions of the sarcolemma, which are called T-tubules. This triggers the sarcoplasmic reticulum, a network of membranous sacs within the muscle fibre, to release stored calcium ions in the muscle cell. The calcium ions then bind to tropomyosin, a regulatory protein. The binding of calcium ions to tropomyosin causes a conformational change, exposing the binding sites on the actin filaments. With the binding sites exposed, myosin, which is bound to ATP, can attach to the actin filament. This generates a power stroke, pulling the actin filaments towards the centre of the sarcomere. As the myosin filaments continue to bind and pull on the actin filaments, the muscle contracts. This contraction is not limited to a single muscle fibre, but involves the coordinated contraction of multiple muscle fibres within a motor unit. The motor unit then contracts as a whole, resulting in the contraction of the entire muscle.